is how blood gets out into the heart from the lungs and how it moves out of the heart to the rest of the body. And so your arrows should flow from the vena cava, which is just above part E, then leaving the heart through part F, coming to the heart through part A, and leaving the heart through the outer. That is the direction of the flow. Question number C is asking, give a reason why the wall of chamber C is thicker than D. We all know that the left ventricle is thicker than the right ventricle. But the question is, why is it? Why is it thicker? Somebody? Why is the left ventricle thicker than the right ventricle? Some answer? Elvis?
This is because the left ventricle pumps the blood to the whole body, while the right ventricle, compared to the right ventricle, which only pumps blood to the lungs, thus the left ventricle needs more pressure.
Okay, that is okay. Now, when you're answering this question, the way he has put it, Elvis, that the left ventricle pumps blood to all parts of the body or to longer distances. Then you have to compare it with the right one, which pumps blood to the lungs. So both of them have to be there. You have to talk about what role the left ventricle plays and what role the right ventricle plays. So if we just talk about the left ventricle pumping blood to the whole body and leave it like that, then you will not score all the marks. Now that was an easy one. I want us to go down and answer the question that was set in KCSE 2015. And this is now where students start struggling. It starts becoming hot. Students are asked to describe how the mammalian heart is structurally. Structural is how it appears and what it has. Now, explain how the mammalian heart is structurally adapted to its functions. I know you've learned before about the adaptation question. Now, the easiest way to learn about an adaptation question in class, if you any structural question, I draw a table. Okay, I will identify. I will have a book for structural questions. In any topic from topic 1 where I get a structural question and I draw a table. The first column will be of the structure. The second column will be on the modification. And the third column will be on the function. So your adaptation question answer, any answer you're giving on adaptation question should have the 3 parts, unless otherwise, because there are some parts that don't have modifications, but most of them will have. So it should have the structure, modification, and what? Function, 3 aspects. Now, how does the marking take place? In most cases, the structure and modification carries the first mark, then the function carries the second mark. It will always have 2 marks. Now, how is the mammalian heart? I just want to get a few responses from you on how the mammalian heart is structurally adapted to its functions. So I want to see hands up. And we move a bit faster because we have a couple of things to cover, and we're moving from the simpler to the complex. Okay, how is the mammalian heart structurally adapted to its functions? I am not seeing hands. Nelson?
The mammalian heart contains a pericardium fluid that secretes, contains a pericardium membrane that secretes the pericardium fluid, which helps in reducing friction.
Okay. Nelson, that's a good answer. But you've said that it has the pericardium that secretes pericardial fluid. Now, my question is, now we dwell as much as possible on structure, modification, function. Now, how is the pericardium adapted to secrete the pericardial fluid? Now, that should be it should be in your answer. How is the pericardial membrane adapted to secrete pericardial fluid? Okay, so that it can be able to carry out the function. Somebody, what do we say about the pericardium? What about it? What enables it to secrete the pericardial fluid? Now, which plays that role? Because leaving it that it has the pericardium that secretes pericardial fluid is not complete. It's not a direct modification. Somebody. What about the pericardial membrane? Somebody. Alvan.
Prevents
Okay.
overdilation.
Not
Yes.
sure about I'm not sure about it, but I think it has the pericardium. It has some glands which
Yes.
secrete the pericardial fluid.
Yes, that is the point. The pericardium. Now, you see, the question structure and modification, it looks very easy, but it has key areas that you can easily fail. It has the pericardium or the pericardial membrane is glandular. Sawa. It therefore secretes pericardial fluid, which lubricates the heart during pumping or when the heart is pumping. Sawa. So the modification about the pericardium is that it has glands. Then now you can add other functions of the pericardium after identifying the modification. Actually, some books will tell you that the pericardium is a transparent membrane that is glandular to secrete pericardial fluid that lubricates the heart during pumping. Okay? Then what are other functions of the pericardium? Somebody? Other functions of the pericardium. Yes. Now you can add them. Okay? As a continuation. I need hands and I want us to move fast. Let us contribute. Yes, I need to see hands. I'm not seeing hands. People are not talking. People look bored and you guys are home. Yes, Akaranga. Akaranga has left. Alvan. Alvan. Am I the one who is not getting you guys, or am I the one who is lost? Alvan, are you there? Yes, you can also chat your responses. By there, I'm seeing a response from Maxwell here that the pericardium checks overdilation of the heart. Ryan is saying that it has a muscular septum to separate oxygenated blood from deoxygenated blood. Okay, that's okay. Can we split points of the pericardium membrane so that we can give all the adaptation of the pericardium membrane as one? The points of the pericardium can be split, but it's important for you to start with the modification one, saying that the pericardium is glandular, secretes this fluid for this role. Then you continue saying, now another mark, after scoring those 2 marks on the structure and modification and function, the first one about being glandular, then you can score by saying that the pericardium keeps the heart in position to prevent overdilation. That one you will score. You will score by saying that the pericardium is surrounded by a thick layer of fat that cushions the heart against mechanical damage, okay, or cushions it against a shock. Then, yeah, those are I think the 3 points. First is about it secreting pericardial fluid, which makes it, and the role it plays. The second one, it's about being able to prevent overdilation. And the third is by the fact that is covered by a layer of fat that cushion it against mechanical damage. 3 of them. I'm seeing the charts here. It does not help. I want you to eliminate in your answering. I want you to eliminate the word help. You know, when you say something helps, and in biological answers, avoid the word help. If it helps, then it means that there is another one that plays the role. Now, because there's no other one, it actually absorbs the shock. Okay, it does not help, and that is the fluid that does that. It has tendons. No, you don't say that it just has tendons. Okay, it has tendons which are inelastic. Okay, it has tendons which it is because they are inelastic. That's why they prevent the valves from turning inside out. Sawa sawa. So you say that the cuspid valves have tendons which are non-elastic. That's the first mark, to prevent the valves from turning inside out during the systole or when the ventricles contract. So you have to talk about what is it that the valves have that prevents the what is it that the tendons have that prevents the valves from collapsing? It is because they are inelastic. Sawa sawa. Yes. Then somebody saying about presence of cardiac muscles. Now, what about the cardiac muscles? What makes it? You know, he's saying that presence. William is saying that they have cardiac muscles that continuously pump blood without getting fatigued. No, that looks okay, but it's not. What is it about the cardiac muscles that prevents them from getting fatigued? So they have special fibers. They have cardiac muscles which have special fibers that enable them to contract and relax rhythmically without fatigue. One of the reasons why they don't fatigue is because of the special muscles they have. 2, it is because they have numerous mitochondria. So you can still say that the cardiac muscles, another modification, is that they have numerous mitochondria to generate energy for muscular contractions. That's the second. So the cardiac muscle, number 1, they have special fibers. And because of that, they're able to contract and relax rhythmically without fatigue. Number 2, they have numerous mitochondria. Sawa. Number 3, that's the third one about the cardiac muscles. You can say now they are myogenic, as somebody is saying here. Myogenic, meaning that they are able to contract and fatigue rhythmically without nervous stimulation. And for that wave to spread evenly in the heart, then these muscles, they are interconnected. Okay? And because of that interconnection, they form a network of fibers called intercalated discs, which rapidly and uniformly spread the contractions. So you can simply say that the cardiac muscles have intercalated discs, which uniformly spread the contractions. So the cardiac muscles, we've talked about them being myogenic, having intercalated discs, or they are interconnected. You can talk about them having numerous mitochondria and them having special fibers. Those are 4 on the cardiac muscles. So You don't just say myogenic muscle, talk about cardiac muscles being myogenic, and they're able to contract and relax without nervous stimulation. I saw a hand from Kibati. Kibati, you had a question or a response?
Kibati? I had a response to the myogenic part of the cardiac muscles.
Well, has it been responded to, or you need some to clarify something?
It has been responded to.
It has been responded to. Are we guys moving together, or somebody's lost? Are we at speed?
Yeah.
Hello? Somebody from the other side getting me?
Yes.
Yes. Now, so we have talked about the pericardium, and we talked about three roles there. We have talked about the cardiac muscle, and we have talked about 4 modifications. Now, whichever structure you talk about, and you talk about the modification and the function, those are 2 marks. So in the cardiac muscle, you can score as many as 8 marks from the cardiac muscle alone. Then other parts now, there are some parts that don't have modifications. For example, the heart having the coronary artery. Because the coronary artery is highly branched, you can say that, to supply the heart with the nutrients, or the heart muscle with the nutrients, you can say that. Or you can still say, you're allowed to say that it has the coronary artery that nourish, supply oxygen, supply nutrients to the heart muscles. You are allowed to use that. But if you want to talk about the modification side, you can say that the coronary artery is highly branched to carry out these functions, supplying nutrients, supplying oxygen, nourishing the heart muscles. Still okay at that point. You can still score independently for the coronary vein by saying that the heart has the coronary vein, which removes metabolic wastes. Still, you can say that the coronary vein is highly branched to collect and remove metabolic wastes from the heart muscles. That will still be very correct. Which part have we not talked about? You can still say you can still say. Now, give me other responses that I have not given, and other responses that I have not given. Somebody raise up your hands. You've been careful. We talked about the pericardium. We talked about the layer of fat that covers the heart. We've talked about the cardiac muscles. We've talked about the coronary artery, that's the blood supply. Which other parts have we not talked about? And it is important that when you're answering this question, you list all the parts of the heart down. Now, please don't list the other parts that are external to the heart. I mean, they are connected, sorry, those are connected to the heart. It's not very okay to list the pulmonary arteries, the pulmonary vein. They are not part of the heart. We're talking about what is in and around the heart. Outside, you have the pericardium and the adipose tissue. We also have the coronary arteries and the coronary veins. Then now we come to the inner side. Okay, you come to the inner side. So Raymond, your microphone, you are on. Maybe you want to say something, Raymond. Raymond, if you have nothing to say, I will mute. Somebody's talking about the vagus and the sympathetic nerves. Yes, those ones don't have modifications. The heart is supplied by the vagus nerve. So you don't just say that it has the vagus and sympathetic nerves. What is the role? What is the role of the vagus nerve? And what is the role of the what? Sympathetic nerves. Okay, you have to mention. So what is the role of the vagus nerve? Does it slow down? Does it speed up the rate of heartbeat? Because the contraction and relaxation of the heart is not under the control of the brain. It is the rate of heartbeat, the rate. So when you talk about the vagus and sympathetic nerve, you connect them to the rate of heartbeat. Do they slow down or do they speed up? Somebody to respond to that. Which one slows down? Which one speeds up? Yes. JP, I'm seeing JP, I don't know who.
Vegas slows down.
And the sympathetic nerve speeds up.
Vegas.
Some people confused. They think that the vagus, the sympathetic, being sympathetic, it should slow down. The sympathetic nerve speeds up. The vagus nerve slows down the rate of heartbeat. You score 2 marks for those two. It has the vagus nerve that slows down the rate of heartbeat, and the sympathetic nerve that speeds up the rate at which the heart beats. Another part that we have not talked about. So it has a solid septum or muscular septum that separates the left side of the heart, preventing mixing of oxygen between the blood. That's very okay. Abdi Malik. The same way, something has been repeated by Maxwell. You can also say that the heart is divided into 4 chambers. It has the atria to receive blood and the ventricles to pump out the blood. That will still be okay. The ventricles, you can still see that the ventricles have thicker walls than the auricles to generate high pressure to pump blood. You can score for that. You can still score when you say that the left ventricle is thicker than the right ventricle to pump blood over a longer distance. Now, when you're doing that, when you're comparing the left ventricle and the right ventricle, or the auricles and the ventricles, use comparatives. Thicker, longer. You getting that? If you say thick, long, you will not be able to score. You compare them. Sawa sawa. Tunaelewana, lakini is somebody getting me from the other side? I'm feeling so lonely in this side here.
Yes.
Are we there? Is somebody or somebody's getting lost? Yes. Are you getting the teacher?
Yes.
Yes, I'm trying to insist on very key parts because this is where issues arise. Then you can still score when you talk about them having valves. Okay, they have valves to prevent the backflow of blood for double circulation, because we have the tricuspid valves and the semilunar valves. They prevent backflow of blood. Or you can still say they have the tricuspid valves that prevent backflow of blood to the auricles. Okay, during contraction, when the ventricles contract, or during the systole. Systole is the contraction phase. Are we getting that? So there are so many points, but as much as possible, my point is, when you are answering an adaptation question, 3 things must be in place. As much as possible, ensure that you identify the structure. So, the left ventricle. Ensure that you identify the modification, thicker. Are you together? And then the reason for that, to pump blood for longer distances or to the whole body. 3 things must appear as much as possible, unless otherwise the part you are mentioning does not have modifications. At that point, if there is any question, I will be ready to respond. I'll be ready to respond. Before now, we move to another question, another area that looks a bit technical. The heart located in the thoracic cavity is not a modification. We are talking about structural modification. How the heart is structured structurally. The location of the heart is not part of the heart. The structure is how the heart looks like. Okay, what is the composition of the heart? What is the heart made up of? How does it appear? So being located in the thoracic region is not a structural modification of the heart. Or adaptation. Modification is how can we define the modification? How it is formed, or how it is structured to carry out a given function. Okay, how is the heart structured to pump blood to the rest of the body? It is by the left ventricle being thicker, okay, so that it develops the force to pump blood to the rest of the body. How is the heart, the muscles of the heart structured or made to be able to contract and relax without nervous stimulation? They are myogenic. Modification is the way it is structured. Something is structured to be able to perform a given function. Can I explain the working of the sinoatria node? The sinoatria node is also called a pacemaker. The sinoatria node is able, ni kama it is hii kitu inapenda kuleta problems. It's a tissue that's made out of several cells. Now, this tissue is generates a spontaneous and rhythmic contraction ability. It's able to generate a wave of contraction and relaxation by itself. The sinoatria node is located just in front of the vena cava in the right atrium. So it generates that spontaneous rhythmic contraction and then spreads it to the septum there, where we have the atrioventricular node. I'm going to explain that in the next, the way now the wave travels in the next question. The atrioventricular node, it acts as a pacemaker. It spontaneously generates a rhythmic contraction, which initiates and maintains contractions of the heart. Or you can say that it is spontaneous rhythmic activity initiates and maintains contractions of the heart. I don't know if we are there, Eric. Eric, that was your question. Eric, are we there? What prevents blood in the outer from flowing back to the heart? Those are semilunar valves. We have semilunar valves at the base of the major arteries. Arteries lack valves. When you're differentiating arteries with veins, you say they lack valves, except at the base of the major arteries. So at the base of the outer, you will have semilunar valves to prevent backflow of blood to the heart. I hope we are there. Are we clear? Are you able to get Mwalimu from the other side?
Yes. You're very clear.
Yes.
Mr. Onami.
Yes.
Yeah, I'm requesting we do a quick roll call at this time. This is 1 hour after the class, and it's the right time for us to do a roll call, please. And, now, for the boys, I would like to advise that we can see a number of boys using very funny pseudonyms. This is an official class, and therefore use your proper name. Don't use initials. I can see names like GKW something. I can see JB. I can see some other funny names. Please rename yourself the correct name, because we're now doing official roll call, and that is the name we attach to your class. So look at your screen right now. You should see a pop-up. Pick your class, submit. You can see it from now. -huh, 50, 80, 100. You can see that. 130. This should take you less than 2 seconds to finish.
Window as a smaller surface area than the column horizontal area.
We still have some who have not. We still have some who have not. I don't know why some students take too long to submit. Submission should be instant. Pick your class, submit. That is it. We are at 156 out of 180, 58, 59. We still have about 17 students who have not submitted their register. At 86%, that's a very small number. We want at 90. Okay, I think we bring it to an end at this stage. So we stop it now. Okay, sir, you may go on. Thank you.
Thank you, sir. So that is the first part. Now we go now to questions that look a bit complex for students. Now, sometimes they look complex because of the way they are presented. Now, one of the questions is describe the initiation. Sorry. There is a question that I may have, but yes. So describe the initiation or describe the heartbeat. That question can be asked in a very simple way. Describe heartbeat in man or heartbeat in mammals. That's an easier one. But then in last year, 2022, they were asked the same question, but in a different manner. This one was describe the initiation and control of the heart. Now that one was a bit direct. Last year they were asked to describe how the mammalian heartbeat is controlled. How the mammalian heartbeat is controlled. And so that was 12 marks. So one of the greatest issues there was most students thought that they will explain how they just present how the vagus and sympathetic nerve control the rate of heartbeat. But we're not talking about the rate of heartbeat here. We're talking about heartbeat as a whole. This is the same question as describing heartbeat. So whatever answer that was needed here was for the student to explain the heartbeat. How does the heart beat? Why is the word control always used when you are talking about heartbeat? It's because the way the heart beats is organized in a way to avoid rupturing of the heart. I mean, the way the waves flow from the sinoatrial node, then down to the ventricles to initiate a contraction should be in such a way that it also gives time for the relaxation take place, so that contraction and relaxation don't take place at the same time. The statement I put somewhere here, if you can see on my screen, the last paragraph, that there is a short delay of about 0.1 seconds before the waves pass from the atria to the ventricles to prevent them from contracting simultaneously. There should be no simultaneous contraction between the auricles and ventricles, because that one will make the blood meet at a central place, and this will be disastrous. It may cause even rupturing of the heart. And so it is a controlled, the heartbeat itself is a controlled process. That's why when you get a question asking you to describe the initiation and control of the heartbeat, or asking you to describe how the mammalian heartbeat is controlled, then you will describe the heartbeat, how the heartbeat takes place. Is somebody getting the teacher from the other side? Is the teacher communicating?
Yes. Yes.
Yes.
Yes.
Yes. Now, for example, now this is how the heartbeat takes place. I'll give you two closely related formula or structures of writing. And so this is a procedural answer. The answer should be flowing. Why? Because it is something that the heartbeat takes place in a controlled manner, so that to avoid if the disastrous thing that may happen if it is not. So that's why your answer should be flowing, and it must start from the pacemaker, the trouble structure. They are called the sinoatrial node that initiates the contraction. So it initiates the heartbeat. After it has initiated the heartbeat. Now don't use SAN. I just use this one so that I can do it fast. So it sends the electrical waves to the atria, making the atria to contract first. Then from there, the waves then pass from the atria to the atrioventricular node. Atrioventricular node is found at the atrioventricular septum, that muscular layer that separates the left and the right side of the heart, preventing mixing of oxygen and deoxygenated blood. That part. Hapo kwa junction ya, let me just come to the diagram here. This diagram here. If you can see here, there is a septum, and I want to tell you where that node is placed specifically. The atrioventricular node. The atrioventricular node is placed almost up katikati, the junction between the ventricles and the auricles. At that point, that's where you find the atrioventricular node. So that's where the wave comes. You must not write this. That's a secondary. It first hits the auricles, making them to contract. But you can avoid that and just simply say that the wave, the electrical wave, moves from the atrioventricular node directly. So not from the atrioventricular, but from the sinoatrial node to the atrioventricular node. Right together, it's picked by the atrioventricular node, then carried down to the Purkinje tissues, which now spread the wave to the ventricles, now to initiate that contraction, because the ventricles are the ones that now pump blood out of the heart. So this is what I'm trying to say here, that the heartbeat is initiated by the sinoatrial node. Then the sinoatrial node starts the electrical waves, and then they are taken up by the atria, which contract, and then the waves are spread to the atrioventricular node. Now, this one is not important. It's not important in your examination, but it's good for you to know that when this process is taking place, there is a short delay, so that to prevent, because if it takes place spontaneously, remember the auricles are receiving blood and then contracting to push that blood down to the ventricles. If the contraction of the auricles takes place at the same time with the ventricles, imagine the auricles releasing blood to the ventricles, and at the same time the ventricles pumping blood up to the outer part of the heart, or to outside the heart. The blood will meet at a central place, and that will be detrimental, or as others. That's why it gives time, so that the blood moves down first, and then the contraction takes place. Then, so the wave moves from the atrioventricular node here, leave this one side, and then it moves down to the Purkinje tissues. Now, these Purkinje tissues are tissues that emerge from where the ventricles start, below the atrioventricular node, and then they spread to the ventricles. So the Purkinje tissue now spread the waves through the ventricles, causing them to contract from the base. That's why if you watch the heartbeat, if you take time to watch a video of the heartbeat, you will realize that the contraction starts below, from below, and then they push upwards. Okay? Yeah. So then now you can come and conclude saying that the vagus nerve now slows the rate. The sympathetic nerve speeds up. Okay. Yeah. So the same question was asked in and it caused a lot of problems last year. Question number 8B. Okay. And this is how it was supposed to be presented. I hope somebody is getting Mwalimu from the other side.
Yes.
Yes. Okay. So can Mwalimu repeat something? Should Mwalimu repeat something? Any question? That's what I mean. Is there any question?
Pardon the procedure.
The procedure is, I hope I have presented something there. In simpler terms, this is it. When you're explaining how the heartbeat is controlled, okay, you start from when the heartbeat starts till it causes an effect in contracting the heart. It's a procedural one, okay? So you state that the heart contracts without nervous stimulation. That's okay. Then the contraction arises from within the heart. Now, what initiates this? The synarteria node or the pacemaker initiates the contractions. Then it sends the wave of excitation to the atrioventricular node. The atrioventricular node sends the waves down to the Purkinje tissues at the bottom of the heart, causing the ventricles to contract. The contraction of the ventricles takes place from the lower part to the upper part. That's just secondary. Then the rate of heartbeat is under the control of the nervous system. Rate, not the heartbeat, the rate at which it takes place. The vagus nerve slows down, while the sympathetic nerve speeds up the rate of heartbeat. So it's you showing how it takes place from the synarteria node to the ventricles. So I don't know if I should repeat again. Is it clear?
Yes.
Yes. So if there's any question, you guys can ask. Now, that is what I thought about the heart. So the systole and diastole, you guys can study at your own time. Is there any question on transport of blood there around the heart that somebody wants to ask before I go to transport of respiratory gases? The second part that causes issues again. Kuna swali? You're free to ask. Okay, I hope Mwalimu is communicating. Then I want us to try to square out on how respiratory gases are transported. Now, there are two respiratory gases. One is oxygen, the other is carbon dioxide. Why are they called respiratory gases? Because one is involved in respiration, that's oxygen. And another one is a byproduct of respiration, that's carbon dioxide. Now, these gases are important. One is transported into the body for respiration, another one is transported out. Now, the transportation of oxygen is easier. I'm not going to touch that one. What gives us problem is the transport of carbon dioxide. This transport can be set in two ways. It can be set in paper 1 or paper 2 as a question structured like this one. Or it can be set as an essay. Now, this question here was in 2017, paper 2, question number 2. The table below shows variations in the form carbon dioxide is transported in the blood at rest, and during a physical exercise. So there's a comparison of how carbon dioxide is transported at rest and how carbon dioxide is transported during a physical exercise. Okay, I want us to look at that. Now, form of transport, dissolved carbon dioxide at rest is 0.52. Not that one. During exercise, it is 0.97. The bicarbonate ion, now nowadays we don't use the term bicarbonate, we use hydrogen carbonate ions. Okay. Now the bicarbonate ion is 12.34. Exercise, after exercise 13.68. There's a rise. Carbon dioxide bound to the protein, at rest is 0.26, at exercise 0.16. Okay. Now, carbon dioxide in plasma is 13.12. You know that carbon dioxide is also transported in the plasma. You know that. That was our form of weak carbonic acid. Then carbon dioxide during exercise is 14.81. Now we ask ourselves, pH at rest is 7.42. Exercise is 7.09. The pH is lowering a bit. Acidity is increasing, as you can see, because you know the pH line. I hope we are getting the question. Have we read the table and understood the table? I want us to understand the table first. That is the most important thing when you're doing a diagrammatic question. Have we understood the table? Can you go to the questions?
Yes.
Yes. Now, the first question is, explain why more carbon dioxide is transported in the form of bicarbonate ion. Okay. Why is carbon dioxide transported in the form of bicarbonate ion? Nowadays, the same question will be asked, and it will be asked like this. Explain why carbon dioxide is transported in the form of hydrogen carbonate ions. So also instead of bicarbonate ions. Okay. Yes. Can somebody answer for me that one? I need an answer in that, please. Somebody, hands up. Dominic. Somebody, a hand up. Why is it that carbon dioxide? Kuna mtu akona jina fani hapa. And please write your name. This guy called Jay. Somebody, Victor. Why is it that carbon dioxide is transported in the form of hydrogen carbonate ion or bicarbonate ion?
To avoid altering the pH of the blood.
Okay, to avoid altering the pH. Not really. That is why carbon dioxide is transported in the red blood cells. So, to avoid altering the pH is an answer for why carbon dioxide is transported in the red blood cells. So, tunaelewana?
Yes, sir.
Yes. So I want an answer. Let me give you the answer.
Yes.
So, it is because now you learned that in the red blood cells, on the cells of the red blood cells, we have some enzyme called carbonic anhydrase. You remember that?
Yes, sir.
It is simply because of carbonic anhydrase enzyme. This enzyme speeds up the conversion of carbon dioxide to weak carbonic acid. So, now weak carbonic acid is not stable. It dissociates. Association is like breaking down. It dissociates into hydrogen carbonate ions. So, or now the bicarbonate ions here. That's why you'll get now more bicarbonate or hydrogen carbonate ions because of the work of carbonic anhydrase. Sawa. Naelewana. Carbonic anhydrase is an enzyme that speeds up the reaction between water and carbon dioxide. Sawa. To form weak carbonic acid. Now this acid dissociates to form now the bicarbonate ions or the hydrogen carbonate ions. I told you nowadays we don't use bicarbonate ions, we use what? Hydrogen carbonate ions. Sawa. Naelewana? Yes. Please differentiate this question with the question of why carbon dioxide is transported in the red blood cells. Sawa sawa, hiyo tunakuja kuianswer briefly. I just wanted to give you the structured question that can be asked outside the essay questions. Number B, somebody read for us number B. You guys are so cool the other side. Ama jamaa wameenda lunch, wanakula tukisoma. Anton, you want to read for us? Victor Mula, tusomee number B.
Account for the high total plasma content of carbon 4 oxide during exercises.
Yes, why is it that there is a high total plasma content of carbon 4 oxide during exercise? Why is it that we are having more carbon 4 oxide in the plasma? Yes, during exercise than when a person is at rest. A hand in the air. Answer that one. Jim Collins in the rank. Is it
during exercise? The cells respirate, producing carbon dioxide.
Okay. Is that one the reason? You see, at a gym, I want you to understand something. At a say, if you are at rest, because say you are using a lot of energy to reason, but when you are at rest, your body is still respiring. It's called basal metabolic rate, respiring normally to sustain the body functions so that you're able to function. So, but then what is the difference when you exercise? This is it, Jim. When you exercise, the body requires higher amount of energy. That's the first part. So, and so because of that, there is increase in the rate of respiration, or there is a high rate of respiration, higher, let's call it higher rate of respiration. And because of that, more carbon 4 oxide is produced. The thing is, what's the difference between at rest and exercise? In both, respiration is taking place, but what's the difference? There is production of higher carbon 4 oxide. Why? Higher rate of respiration. Why? Higher amount of energy is required. Tuko sawa? Jim and the rest. Tuko sawa?
Yes.
Yes, sir.
So that is another part that you need to understand. Know how to differentiate. Know how to compare. Use comparatives. That will be important. So the essence here, are you able to identify it's more, it is higher? Sawa. That is where the issue is. That's where you may miss marks. Not just writing. If you write plainly, you will not score all the marks. Now, state how one's involvement in an exercise affects the blood pH. Now, what's the relationship between exercise and pH? What's the relationship? Alvin in Europe, what's the relationship? That when you engage in a very tough exercise, there is an effect in your pH. And as we have seen, that the pH moves to acidity. You look at the blood, it's moving from 7.4 to which is more alkalinic. Some acidity is added, lowering the pH a little bit. What is the relationship? Okay, somebody, hands in the air. Ryan Mirur.
During intense muscular activity, blood is unable to supply enough oxygen for aerobic respiration to generate all energy required for contracting muscle cells. So anaerobic respiration occurs in the muscle, resulting to production of lactic acid. Therefore, the lactic acid accumulates in the muscles.
Okay. Okay. You're still okay. The only part you got a little bit off guard is when you started talking about fatiguing the muscles. We're not talking about fatiguing the muscles here, Ryan. We're on the pH side. You need to understand the aspect of the pH side. You're okay when you started saying that during a physical exercise, so of course, more carbon dioxide is produced. Why? Because there is a high rate of respiration. Now, let me tell you something. Joram, I can see your hand. Yes, please.
Teacher, I think during exercises, energy is needed, and therefore respiration rate is higher. Hence, carbon 4 oxide production will obviously increase, and that will cause a drop in the pH in blood.
Okay, that's still okay. Mentioning carbon 4 oxide will be very okay. Mentioning lactic acid will be very okay. Sawa, Joram. It's true that during a heavy physical exercise, there is a high rate of respiration, and high rate of respiration is during normal cellular metabolism. A lot of high rate of respiration takes place. More carbon 4 oxide is produced. Sawa. Now that one lowers the pH. Or you can say that during heavy exercise, there is high rate of respiration. More lactic acid is formed. Sawa, which now lowers the pH in the blood plasma. Now we know why do we form a lot of lactic acid and a lot of carbon 4 oxide? It's because when you are engaging in a heavy physical exercise, what happens is that the body is not able to respire aerobically. Aerobic respiration takes a longer time. So the body resorts to anaerobic respiration to produce energy faster. Now, in the process, you know that anaerobic respiration produces a lot of carbon 4 oxide because it uses less oxygen. More carbon 4 oxide is produced. You remember that experiment, an experiment that we always do, where I think you have been asked before, you have lime water on the other side, then you have an aerobically respiring yeast the other side. You're told the observation. You'll observe a lot of precipitate when a tube is connected from anaerobically respiring yeast to lime water. But when you connect aerobically respiring yeast to lime water, you'll see less precipitate because aerobic respiration produces less carbon 4 oxide. That one should be in your mind. And at the same time, anaerobic respiration produces lactic acid. Lactic acid further lowers the pH. Sawa. Tunaelewana lakini somebody's getting mwalimu from the other side.
Yes.
Is somebody lost the other side?
No.
Okay. So we have understood. The thing is that I want you to understand this. Tuko sawa? Now, I hope so.
Yes.
I hope so. If we are together, somebody read for me question and D. Somebody read for me D. John Paul.
Name the protein responsible for the transport of carbon 4 oxide in blood.
Which protein is responsible for transport of carbon 4 oxide in the blood? Somebody respond. John Paul, you can respond to that. Which protein? Collins, Kibadi.
What about hemoglobin?
What is the answer, Kibadi? Is it hemoglobin or what about it?
It's hemoglobin.
It's hemoglobin. Yes, hemoglobin. That's okay. And this is the framed question, structured question. Now, if now we go down and ask ourselves here, if you're asked now to describe how carbon 4 oxide is transported in the blood, okay, how will you answer it? If you're asked to describe carbon 4 oxide from the tissues, how carbon 4 oxide is transported from the tissues to the lungs for excretion, how does that process take place? What about it? Okay. How do you answer that question? Somebody. Because there are key things that you need to understand. Somebody to help me there. In what forms is carbon 4 oxide transported in the body? That should be your first thought. In what forms are carbon 4 oxide transported in the body? Sawa. And how do these forms come about? Okay. Somebody to respond to that, somebody to help me understand. Tell me the forms in which carbon 4 oxide is transported in the blood. Hands in the air. No hands. Still, I can't believe. Michael Gitonga. Michael.
In the form of hydrogen carbonate ions.
Hydrogen. That's the first one. Hydrogen carbonate ions. Another one. Alvan.
In the blood plasma.
In form of what? The blood plasma, Alvan. We have to say that one of the forms in which carbon dioxide is transported is in the form of hydrogen carbonate ions, okay? But permanence. Number 2.
Carbamino acid.
Carbamino acid, not carbamino acid. Kibadi. But I'm not able to get you, and you mute, please. Yusuf.
In form of weak carbonic acid in plasma.
Weak carbonic acid in the plasma. That's very true. The third one.
Carbaminohemoglobin.
Carbaminohemoglobin. Now, when you're asked how, then you identify those three. You have to know them. Now, your task here is after knowing that, hey, one of the ways is in the form of hydrogen carbonate ions. Number 2 is in form of carbaminohemoglobin. Number 3 is in the form of weak carbonic acid, but in the plasma. Now, your role is, how do I scientifically explain this? Okay. Now, after knowing that, then after asking yourself that question, the next question is, how is it converted to these forms here? What necessitates the conversion? I've already talked about that. What necessitates the conversion? You've talked that one in the previous question. What necessitates that? How do you start answering it? Somebody, just give me a prelude. How are they converted to these forms? Now, if this part is giving you a problem, I want you to go and look at principles, the new principles of biology for volume 1 and 2. It has done very well in this, very well. You will understand it very well. Now, somebody to tell me, how do you answer this question? Is somebody getting Malim from the other side? Oh, Malim is talking by himself.
Yes.
Okay.
Now,
yes. Let me try to bring it down. Now, the first thing you start, if you just ask a question on how it is transported in the blood, just like that, then you will start by how this carbon dioxide is fixed into the red blood cells, because we say that it is important for carbon dioxide to be transported in the blood for 2 main reasons. Okay. The main one is to buffer the pH, prevent the pH from becoming very acidic. Because if the plasma becomes acidic, plasma is what it surrounds our cells in the body. Then you know what the effect of that the cells. Most of the cells are made of protein. They'll be denatured. There'll be a lot of disaster in the body. You may die. And so the essence here is to buffer the pH. That's why it's not in the blood. I'm going to explain all that pole. So the first thing that happens for that binding to take place, and you should remember that 85% of the carbon dioxide is transported in the blood. 85 to 95% transported in the blood. Now, bulk of it is transported by in the form of hydrogen carbonate ions. How? Enzyme carbonic anhydrase. That's you start from carbonic anhydrase. Inside the red blood cells, it speeds up the reaction between carbon dioxide and water. And when this happens, it forms weak, not just carbonic acid, but weak carbonic acid. Weak carbonic acid is unstable. it easily dissociates to form hydrogen carbonate ions and hydrogen ions, two ions, hydrogen carbonate ions and hydrogen ions. Now, these hydrogen carbonate ions, they quickly diffuse out of the red blood cells to the plasma, where they are carried through the lungs. Hydrogen ions bind with hemoglobin in the red blood cells to form hemoglobinic acid. Now, that one is, they are transported in that form. Because if for lungs. If those hydrogen ions leave, sorry, I'm saying that this, I'll say that carbon 4 oxide reacts with the water under the catalyst called carbonic anhydrase to form weak carbonic acid, which is unstable. It easily dissociates to form hydrogen ions and hydrogen carbonate ions. So now hydrogen carbonate ions will diffuse out of the red blood cells into the plasma. The hydrogen carbonate is not harmful, does not have any effect to the pH. It's transported by the plasma to the lungs. But hydrogen ions, because you know that hydrogen ions now determine acidity. When there are many in a place, they increase the acidity of that. Now they are buffered. They are bound to hemoglobin. They're bound to hemoglobin to form weak hemoglobinic acid. Weak hemoglobinic acid. Okay. Now, this hemoglobinic is transported with the red blood cells to the lungs. Now, if we are asked a question, how the pH is buffered, or how hydrogen ions are buffered, they react in the red blood cells. The first thing is that they react with hemoglobin in the red blood cells to form hemoglobinic acid, which is weak hemoglobinic acid, which is transported to the lungs. Okay, sawa. Naelewana?
Yes, sir.
Then number 2, you should note. Now, you know, once, okay, let us leave that one as that. Then some of the carbon 4 oxide will diffuse into the red blood cells. Now, it will react with amino acids in the red blood cells. And it will form carbaminohemoglobin. It will move out of the red blood cells. It will move out of the red blood cells, combine with the amino acids of hemoglobin. Combine with the amino acids, not just amino, but amino acids of hemoglobin to form carbamino. That's why it's called carbaminohemoglobin, because it's carbon 4 oxide combining with amino acids of hemoglobin to form carbaminohemoglobin. So, which is transport the lungs for excretion. Are you getting Mwalimu from the other side?
Yes.
Yes. Then
yes.
Now some carbon 4 oxide, some carbon 4 oxide will dissolve in the blood plasma. And once it dissolves in the blood plasma, it will form weak carbonic acid. It will form weak carbonic acid. Which may be directly transported to the lungs like that. Which may be directly transported to the lungs in form of weak carbonic acid. Or because of the weakness of weak carbonic acid, it will dissociate or break down into hydrogen carbonate ions, number 1, and hydrogen ions. Okay, hydrogen carbonate ions are transported in the plasma to the lungs. Hydrogen ions, they will combine with the plasma proteins. Hydrogen ions cannot just be left like that. They will interfere with the pH of the blood seriously. That will kill the cells. So it has to be buffered. How do we buffer it in the plasma? They react with plasma proteins to form proteinic acids. Proteinic acids, they bind the hydrogen ions. They prevent them from altering the pH of the blood. Are you getting that? So they form proteinic acids, which are transported now to the lungs. I don't know if somebody's getting me from the other side. Is somebody getting me from the other side? Now people are hungry.
We're getting.
Are people dying of hunger from the other side?
Yes. Yes.
Is getting low. Glucose is getting low. Ryan?
Yes.
Glucose is getting low.
Yes.
Just be patient a bit. So we tackled some of these issues and then you guys do go. Nelson, I'm seeing a hand up.
Do those acids dissociate when they arrive at the lungs, like the proteinic acids and do the acids, like the proteinic acids, dissociate when they arrive at the lungs?
I'm coming there. That's a good question, Nelson. I'm coming there. I first want you to understand how the packaging takes place. So, both in the red blood cells and in the plasma. So, tumeelewa hiyo? How the packaging takes place. Yes. Until we form both hydrogen carbonate ions and hydrogen ions in the red blood cells and hydrogen carbonate ions and hydrogen ions in the plasma. Have we formed that? Then we now buffer the pH. How do we buffer the pH? In the red blood cells, we buffer the hydrogen ions. We buffer the hydrogen ions by reacting them with hemoglobin to form hemoglobinic acid. In the plasma, again, we buffer the hydrogen ions by combining them or by reacting them with plasma proteins to form proteinic acids. So we asked two ways of buffering the hydrogen ions. I think now you know. So. Then now, all of these are transported into the lungs. In the lungs, the hydrogen is released. It combines with hydrogen carbonate ions. Are you together? To form weak carbonic acid. Now, weak carbonic acid now releases or dissociates to release carbon dioxide, which is now excreted out of the lungs. It is now excreted out of the lungs. Sawa. Yes. Now that's how it takes place. Okay. And this one, in the excretion process, it takes place diffusion. If you remember diffusion, because of high concentration of carbon dioxide in the blood, less in the alveolar space. So carbon dioxide diffuses into the alveolar cavity, oxygen diffuses back into the blood. Kuna swali hapo. This is the area where I expect a lot of questions. Please do ask. Now I'm asking a question, sawa, and I'm expecting an answer, a 2-mark question. Now explain why it is important to transport carbon dioxide in the red blood cells and not in the plasma. What is the importance of transporting carbon dioxide in the red blood cells and not in the plasma? Why is it important? I expect answers. So I've also not eaten anything, so we are together in this. Somebody, why is it important to transport carbon dioxide in the red blood cells and not in the lung? Somebody in the plasma, somebody asked the same question when somebody answered this question, the previous one. Now you carry it forward. Somebody respond. Somebody respond to that. Sakawa. I'm seeing John Paul trying to respond. John Paul, are you responding?
Yes.
Yes, respond.
It is faster due to the catalytic effect of enzyme carbonic anhydrase, which facilitates transport of carbon dioxide in the red blood cells.
Okay, now that's the first one. Very important. It is because within the red blood cells there's carbonic anhydrase, which is used to speed up the loading and offloading of carbon dioxide. Yes, carbonic anhydrase speeds up the offloading, the loading and offloading of carbon dioxide. Number two, somebody give me the second answer. Alvan, I saw your hand in the air.
The pH of the blood is maintained.
So the pH is maintained. It buffers the pH. Buffers the pH, maintains the blood. The pH is not altered. Oh, those are right answers. Are we clear with that? So I want to give you an assignment in that. Now this is, you have to include more detail. Now the assignment will be, you describe, let me just, how carbon dioxide is transported from the tissues, the lungs, for excretion. If you mind, you can inbox me with, I help you see if you've done the right thing. How can dioxide transfer from the tissues? So you have to talk about how it is reformed in the tissues, released, combines with the blood, transported along different blood vessels, until it reaches the lungs for excretion. Tuko sawa? Are we together?
Yes.
So,
yes.
Guys, we still have one small milestone. Can you yawn if you are yawning? A very small one, very little any time here. I want to ask you a few questions. Okay. Now, I want us to agree on one thing here, because time is gone, and I have two key things to cover. I want to cover two things. I want to cover cell division and fertilization in plants. Which one do we go for first? Is it fertilization in plants or cell division? Because I'm seeing that I have to cover one, can't cover both. Which one do you guys have more issue in?
Fertilization.
Fertilization.
Cell division.
Cell division. Okay. Let us
Cell
start with
division.
fertilization. Cell division.
Cell division.
Yes.
Cell division.
So we start with cell division. So in plants, almost in reproduction is key. If you look at the trend of setting, the questions are always there every year. So production in plants, issues with cell division can come, issues with sexual reproduction can come. It came one day about spore formation, and it ate people's heads. Issues with dispersal, tuliopati isi ya dispersal this time around mkakanyaga nao na miguu wengi wenu. Double fertilization, issues that are constantly set. Now, I want us to look at a few scenario, of cell division. Just trying to remind yourself, are you able to identify the type of cell division here? For example, look at question number 35. That was last year, last year's question. It was number 11, A and B. It was a number 11 question in paper 1. Now, can somebody read for us that question? See, people are moving really. Eric, read for us number 35. There.
The diagram below represents a stage in cell division. A, name the stage of cell division illustrated. Give a reason for your answer.
Will you please answer for us that question?
Pardon?
Answer for us that question. Who can answer for that question? Who can answer for that question? I need hands in the air. Kelvin, keep quiet. Keep quiet.
Yeah.
Yes. Aha. Who is this? John Paul has talked. Kevin has got lost. Timothy Chacha. Chacha.
Metaphase.
Metaphase. Just metaphase like that. We have two phases of cell division. Yeah?
Late metaphase.
Late metaphase.
Metaphase 1.
Metaphase 1. Okay. We have phases. We have mitosis and the meiosis, types of cell divisions, mitosis and meiosis. So, and so you need to understand if it is mitosis or meiosis. Now, one thing you need to understand is that in mitosis, if it was in metaphase, you will not see a pair. You will not see a pair of chromosomes along the equator like that. Let me try to show you something here. I know some people might have left by this time, but there are those who want to listen. And I will spend my time fairly well. If it is mitosis, are you seeing my screen? You able to see my screen?
Yes.
Yes.
No.
Yes.
If it was mitosis, hey, you will see them, Kevin, like this, aligned at the equator, not in pairs, singly, chromosomes aligned at the equator, chromosomes aligned at the equator. But when you move down to meiosis 1, you don't talk about chromosomes. You talk about homologous chromosomes. Homologous chromosomes are chromosomes that exist in pairs. Okay, so you'll find them associating themselves in pairs until they are separated. So that's why we don't just say it's metaphase, but we say that it is metaphase 1. Sawa. Naelewana, Kevin, and everybody else.
Yes.
Yes. Now give a reason. Why have you said that it is metaphase 1? Somebody who answered. There's somebody who responded very fast from their hand. We try. John Paul.
Yes. So the homologous chromosomes are still bivalent and they are arranged at the center of the cell.
So they are, you see, in metaphase. No, this association starts. It starts from prophase 1, and prophase 1 is the most important stage in meiosis. Because in meiosis 1, homologous chromosomes will lie side by side. Homologous chromosomes are chromosomes that exist in pairs. They are similar in structure, but may be different in genetic constitution. So in prophase 1, homologous chromosomes lie side by side. When they lie side by side, they form bivalence. That process of lying side by side is called synapsis. And so here, as you can see, they are still side by side. So bivalents are still existing. It is true, the way John Paul has said, that homologous chromosomes are still associated as bivalents. And are at the equator of the cell. That's very true. Another response, another reason. There are other general reasons there. During metaphase, I want you to know, during metaphase of mitosis or meiosis, you realize that spindle fibers are fully formed. At any metaphase, spindle fibers are fully formed. And then if you look at it keenly, the nuclear membrane has completely disappeared. It has completely disappeared. Those are general ones that apply in all metaphase processes. In any metaphase, spindle fibers are fully formed. In any metaphase, mitosis, meiosis, nuclear membrane has fully disappeared. And it leaves chromosomes free in the cytoplasm. Tuko sawa hapo? Kuna swali? Is there a question? Is there a question? Hakuna swali. Now, I give you which one do we do next? Which one? Number 12. Who can do for us number 12? It is now a stage after what we have just learned. Number 12. Somebody to read for us. Who is that? Number 12. And we don't get somebody to read, we stop. And I see the sugar has gone down for most of us. Gavin, Trevor. Trevor, we are not able to hear you. Who can help Trevor? Not able to hear Trevor. Somebody.
The diagram below represents a stage during cell division. And identify the stage of cell division.
Now answer for which stage of cell division is that?
Anaphase 1.
It's called anaphase 1, right? Yes. Give reasons for your answer. Give reasons for your answer. Give reasons. Why is it? Somebody can answer that. Why is it anaphase 1? Why is it anaphase 1? Yes, Victor. Victor?
I can see that the chromosomes are moving to the opposite poles of the cell.
So chromosomes are moving to the chromosomes are migrating or moving the opposite poles, right? That's okay. That's okay. Kibadi? Kibadi?
I would have defined it as
yes
separation of two homologous chromosomes.
I'm not getting you, Collins.
There is separation of two homologous chromosomes from the equator.
Yes, okay. But the answer is correct. But even you should not say two homologous chromosomes. So homologous already means that there are two. So homologous chromosomes separate at the equator. So structure M, of course, the spindle fiber. Now I want to stop from there. If we had more time, we could do double fertilization. So what we had, and also look at what is the other way in which questions can be set when it comes to fertilization. But now time is limiting. So time is limiting, and the sugars are going low for most of us. So will you allow us to stop from there for today?
Yes.
Yeah?
Yes.
And I hope we have learned, right?
Yes.
You guys enjoy your holiday. Sawa.